Okay, so that's kind of your standard fluorescent protein. I wanted to just highlight a few other potential applications with fluorescent proteins. And these are something called a split GFP, photo-convertible fluorescent proteins, and also FRET biosensors as well. So we'll go through those over the coming slides. Okay, so split fluorescent protein, this is where we actually split the sequence that codes for the fluorescent protein over two different molecules. So I mentioned that the GFP molecule is made up of beta sheets, and these form that beta barrel structure. So in this case, we would express, this is our protein of interest, and this is tagged to just one of the beta sheets from, let's call it the GFP molecule in this instance. We can also express the GFP molecule here that's lacking this beta sheet, so that when these two molecules come into contact, we get fluorescence. When they don't come into contact, then you don't get fluorescence. So this can be very, very useful for looking at trafficking of molecules to certain compartments. Say you only want this to fluoresce when it reaches the mitochondria, then you can tag this to a mitochondrial protein, and therefore they will only come into contact when they both are in the mitochondria. This is very useful because this is a much smaller tag than attaching the full GFP molecule on there. So it's only 15 amino acids, and that's what we call an epitope tag, which we'll cover in a few slides time, which is a very, very small amino acid sequence which we attach to our molecule of interest. In terms of photoconvertible fluorescent proteins, so these are molecules which can switch fluorescence from one wavelength, emission wavelength to another. And these normally require activation, usually with something like a 405 nanometer laser. So in this case, these are examples. So EOS, MEOS is a protein which is regularly used for this. Again, the fluorophores forming from the amino acids within the sequence. In this case, it's emitting in the green channel. If we shine ultraviolet light, this causes a structural rearrangement in the fluorophore, and that's switching the fluorescence wavelength that it's actually going to emit in. So we can start off with molecules emitting in green, and that can transition across into a red wavelength. And this is very, very useful, again, if you're doing tracking through a cell, that you can, selectively activate one region of the cell and watch where molecules from that, how they traffic out to different regions. One other common use of fluorescent proteins are in what we call FRET biosensors. So that's Fourier resonance energy transfer, or fluorescence resonance energy transfer. In this case, again, we're looking very often at protein-protein interactions. So two fluorescent molecules that are regularly used for this are enhanced cyan fluorescent protein and an enhanced YFP, so yellow fluorescent protein. So in this case, we're looking at the binding of the substrate to the sensory domain. And what we're doing is we're exciting the cell with a laser which will excite the CFP molecule. When these two molecules aren't interacting, that will create the emission coming out from the CFP molecule. So here we've got a lot of emission coming out from the CFP molecule. When the substrate and the sensory domain come together to form a complex, this brings the YFP molecule closer to the CFP molecule. And rather than the CFP becoming excited and emitting its own wavelengths, it actually transfers the energy to the YFP molecule, exciting that because it's of the correct wavelength to excite the YFP molecule. And then we can collect out the emission from the YFP molecule rather than the CFP molecule. So here in our spectral analysis, you can see the emission from the YFP molecule has gone up in comparison to this case. So it's a really good way of looking at protein-protein interaction. In this case, the fluorophores must be within about 10 nanometers for that energy transfer to occur.